Voice of the Sea, learning from experts across the ocean.
Welcome to Voice of the Sea.
In this episode, we're investigating where Hawaii's fish come from with Dr. Brian Bowen's research team at the Hawaii Institute of Marine Biology on Coconut Island. Brian's team has just returned from a trip to Karismas, a remote coral atoll 1,000 miles south of Oahu that is also known as Christmas Island. While at Karismas, Bowen's team gathered genetic samples from local fish to compare with Hawaii fish. The relationships in DNA that they discover will tell us about the resiliency of our own local fish populations and reveal secrets of the historical migration of reef fish across Oceania. We start off talking with researcher Michael Hoban. So you just got back from Christmas Island.
Yes, it's like nowhere I've ever been.
Tell me a little bit about the island.
So it's an atoll, just like basically it used to be a high island, but it's subsided, and so now it's flat. I don't think any part of the island is more than 4 or 5 feet above sea level. Turns out it's the largest atoll in the world by landmass at any rate, which I didn't know before going there. Which is really cool. It's about 2 degrees above the equator, I think, in the northern hemisphere. There's a large lagoon in the middle, and then the outside there's fringing reefs all the way around that kind of slope down into the abyss. So there's some interesting things to see when you're out there diving. And yeah, it was just great. It's historically not inhabited, so it has there's some evidence of Polynesians having stopped over there, in the 14th century between Tahiti and Hawaii. But there's no real permanent settlement there until the late 19th century. It had changed colonial hands several times, and then a Frenchman leased the island to plant coconuts, and he planted coconut palms all over the whole thing, and then imported a bunch of people from the Gilbert Islands in the Western Pacific to basically maintain the coconut plantation and to produce copra, which is dried coconut meat for oil. And so these people from the country that's now known as Kiribati were brought over there and have essentially stayed there ever since. And they're a culture that Kiribati is a series of atolls as well, so it wasn't a big shock for them to live there. So it's sort of this far eastern outpost of the nation of Kiribati. I think that country technically, based on how far spread the islands are, has the most area that the country encompasses, but in terms of actual land area, it's the smallest, because it's these tiny little islets that are part of, that are separated by vast stretches of Pacific Ocean.
How do they get fresh water there if there's no high island?
They have trouble with fresh water there. I know that, when it rains, you get, essentially it's called a freshwater lens, so you have a water that sits on top of the salt water on these atolls, but they don't have a lot of soil. It's mostly this kind of broken ancient coral, so water does soak through. They have wells, some wells, mostly rainwater catchment, and I think they actually ship in some water sometimes because they'll go through drought periods periodically. So it's touch and go. And then also, of course, with climate change, because they're so close to sea level, as you get more sea level rise and you get more frequent king tides and stuff like that, it inundates the freshwater supply, and so there's not a lot of room for growing much there aside from coconuts.
How was the underwater habitat there?
It was really beautiful, but also kind of sad. So the reefs are almost entirely dead right now there, but they died so recently that they still have all of their complex structure. So it's this, amazing big complex coral reefs, but it's sadly all dead coral. The flip side of that is that there's still a ton of fish. It kind of shows you that the things that we think of as coral reef fish don't necessarily need coral, they just need reef. But there are signs that it's recovering. And so if they get a few years without another nasty El Nino, which is what hit them in the first place, I think that their reefs will come back, because there's a lot of the signs that you see of, you see baby corals growing, and there's a kind of algae that grows over things that is good for corals, and there's a lot of that kind of algae. There's a lot of big upright algae, which is not necessarily good, but there's tons and tons of herbivorous fishes which tend to eat that stuff. So it's not, it's sad to look at, but I don't think it's a hopeless cause. But they're very close to the equator, so anytime they get hit by any kind of thermal anomaly, hot temperatures over time, they get it the worst. And they're right in the path of that hot water that sloshes across the Pacific for El Niños. And so that's what happened to them. They had a warmer than average year, and then El Nino hit. So it just made the surface water temperature be extremely warm for 9 or 10 months or something like that. And this was all in 2015 that happened. But otherwise, yeah, it was beautiful. And there's, things that you don't see here as often, big milkfish and big trevallies and tons of manta rays. There's fewer people, so you see more fish basically. In Hawaii, you don't, especially on Oahu, you just don't see a lot of that stuff because it's fished out. So it's cool to see.
Can you tell me your favorite part of working on this type of project?
I like going into the field, which is probably why most of us got into doing this, right? We like to go diving, we like to look at fish and that kind of stuff. So I love that part of it for sure. I think in terms of the whole picture, when you see something unexpected or you see something that very closely lines up to what you did expect, right? If you're right about, that's always a good feeling. But then there's also, one of the things that I have going with one of my projects is I'm studying these little fish called blennies that are these adorable little things that sit on coral reefs, and they have a little things, yep, and they have a big smile on their faces, But they're distributed very broadly across the Pacific, this one species. But when I looked at the genetics of them, one group of them grouped out way over here, despite them all being nominally the same species. And that group was from the Marquesas Islands, which, like Hawaii, many of the species in the Marquesas occur nowhere else. And sometimes what you get in those situations is you get a species that will diverge genetically, but not obviously to look at. So it'll look like the same species, but it'll end up being something very different. And so I think, I'm still working through the details of that, but I think we call that a cryptic species. And so I think what I have is a cryptic species of this blenny that occurs in the Marquesas. So that was really cool to see.
The University of Hawaii's Sea Grant College program focused on Hawaii's coasts and its communities through sustainable development, safe seafood supply, sustainable coastal tourism, hazard resilience, and healthy coastal ecosystems. Hawaii's Sea Grant.
Welcome back. We're talking about the genetic connections between fish populations in Keras Mas and Hawaii with researcher Joshua Kopis.
To me, the Christmas Island Hawaii connection is interesting because I'm looking at whether or not sea level has any effect on the genetic signal we see between the locations, and whether or not that's different than the shallow coral reef fishes. My dissertation really focuses on what we call mesophotic fishes, which are fishes that live fairly deep, below 30 to 40 meters, where light starts to get filtered out in the water down to 100 plus meters, depending on where you're at. The bottom of the mesophotic is where the light pretty much disappears. So I do a lot of technical diving to get down to those depths and study those fishes. When you get down to 80 meters or more, you start to find a really unique community of fish that are related to the shallow coral reef fishes, but are definitely different species. And we're actually finding quite a few new species of fish on those reefs. Everywhere we go, we tend to come back with a half a dozen new species. So it's pretty exciting. Sea level changes at approximately 100,000 year cycle at approximately 100 meter amplitude. And so when the sea level drops 100 meters, much of the shallow reef is gone. It's dry land. But the fishes that can live below that may not be impacted, and you may see a different genetic signature.
So you're looking at really long-term relationships if you're going back 1,000 years and looking at past glacial periods?
Well, I really am looking at the last glacial period, and really that's a very short amount of time in the scheme of species relationships. Glacial cycles currently operate on about 100,000 year cycle, where the sea level drops during the glacials when we're in an ice age, and as the ice melts and the sea level rises, the bathymetry or the habitat changes. The amount of available habitat for shallow reef fishes increases as the sea level rises. So the sea levels, we're at the top of one of those cycles. Sea level is now higher than it's actually been for quite a while due to other factors like global warming. And it's possible that the connection between Christmas Island and Hawaii might actually be more obtainable at the lower sea level, where the sea levels are 100 meters lower, and some of those seamounts that are now in too deep a water to support any of the coral reef fishes or invertebrates might actually be accessible to them during the LGM or as sea level drops. So that might provide more stepping stones into Hawaii than we currently have.
So when we sort of look at our Pacific Ocean basin now, we don't see all of the stepping stones that might historically have been there.
Possibly,
That's
yes.
really cool.
And that's part of what I hope to resolve in my dissertation.
The Curriculum Research and Development Group in the College of Education at the University of Hawaii at Manoa. CRDG's training roots go back over 40 years. Through professional development programs, curriculum workshops, research on teaching methodology, individualized school and district training, and so much more. The Curriculum Research and Development Group, improving schools, improving education. CRDG.
Welcome back. We're talking with Expedition Collection Leader Derek Kraft. Derek is not your average spearfisher. Most people spearfish for food, but Derek spears to collect samples for DNA analysis, often searching for very small, hard to see, and hard to catch fishes. What is your role in organizing or being part of this expedition to Kiribati?
I am a research assistant actually under my lab mate Richard, and I handle the field side of most of this research. So I'm actually out collecting fish on a weekly basis. I kind of feel a little bit more in charge of a lot of the equipment, so the spears and the bags and the actual logistics of operating what we kind of do day to day. So making sure the spears are sharp, making sure we have vials, and making sure all of our stuff shows up.
How many dives do you think you went on while you were there?
We did 12 in 5 days.
How many specimens did you guys collect?
Probably about over 800.
Across a spectrum of fish, invertebrates.
Yep. We got
corals.
Fish, corals, we got calories, snails. On the first day alone, I think we got about 280 fish. This is our field shed. Hiding up here are some of our utensils. This is a pretty standard pole spear, Hawaiian slings, 3-prong. Just pull the bands forward, and that's loading it. And then if I let go of the spear, the band will fling the spear forward.
Right, and you guys are trying to hit sometimes pretty small fish.
Sometimes they're about this big. And when they're this big, hopefully they're in schools, and then you kind of shoot at the school.
And so you're gonna spear the fish, and then what do you do with it?
You'll have a collection bag with you. This is a fine collection bag here. If you spear a fish, if it's up against something, like a rock or in the sand, that's handy because then it can't get off the spear, and you'll swim down with your net, pull the spear up, put it right inside the net, close it, and then that's how you get the spear off. And now it's in your bag.
What are some of the concerns that you have, say, prepping for a trip, making sure all the gear is gonna get there?
Making sure it all gets there straight. Like these can bend pretty easy and get bent while traveling. Making sure we have enough extra bands is important. These bands will break all the time. We'll usually carry a couple on each dive. making sure we have extra tips, making sure we have files to sharpen them, and making sure we don't poke each other while we do it.
Right.
Most of the times anybody gets poked, it's on land when people are getting ready, not paying attention, and then poke their buddy.
And then what kind of a solution do you put the fish clips in, the fin clips in to preserve them?
It's DMSO, which is a salt buffer. So the tissue can go in there and it'll preserve it, or we could use ethanol, alcohol.
So sort of like pickling something, I guess.
Sure.
Or,
Yeah, but it's a solution that's safe and it'll keep the DNA intact. It's been a real treat to be able to go out into the water a couple times a week or once a week at least. For a lot of people, the field work is, the kind of the fun part and the nice part. You gotta go out, you gotta go free diving, you gotta go scuba diving, you gotta go out on the boat. And to be able to do that part of the work, for most of my job is what I'm supposed to do is a real treat. I get to know the reef really well. I get to know my specimens really well, how they swim, where they're at, how to find them, who's going to be where, depending on what fish we're looking for that day. So to kind of get that more naturalist knowledge out on the reef, I think is a treat.
what are some of the trickier types of fish to get?
Small ones. The small ones, the chromis, we had a really hard time with. Luckily they're in schools and they'll kind of hang out just above all the reef. And so you shoot at the outside of the schools to get them to compact a little bit more, and then as you shoot, you just hopefully get one.